All right, so we're going to continue with classification, and now we're going to look at other molecular techniques that are used. One common technique that is used now a lot is DNA hybridization. So in this technique, what we do is we take two organisms, extract their DNA, then the DNA is heated up so that the DNA pulls apart, and so you have DNA broken into pieces, and that happens for both organisms. So this organism's DNA is pulled apart, and then the organism's DNA is mixed together. And then what we see is there pairing, between them. So if we were to have A, T, C, C, A, G, A here, and over on this strand, we ended up with a T, A, G, C, A, C, T. What we'd find out is like, okay, that matches up, that matches up, that matches up. They're the same nucleotide, but they don't pair up. That means that they're different here in that sequence. So we would find that there's a certain number of nucleotides that pair, and the strands actually match up together, and there's a certain number that don't pair up. Okay. So in that case, we would then calculate a percent match. And what we find is that if it's a 70% or greater, so 70% or greater, we have the same species. And if it's 25% or greater, it'd probably be the same genus. So let's say there's more to it than that, but this is the basic idea. You take the DNA, extract it, pull it apart, try to put the two of them together, see if they match up. Do they hybridize? And if they hybridize really well, they're probably the same species. If they don't hybridize really well, they're probably not the same species. So if we were to take those two types of analysis, the hybridization, and so this would say be hybridization, and sequencing, so the SSU ribosomal RNA sequence, and compare them. What we're going to have is for the single-stranded RNA sequencing. So for sequencing, our rule there is it need to be a 97% match to be the same species. So 97% has to be the same. So let's say here this is only 50%, 25% here. And here's 25, say 50. Now we got our 70% here, 90%. If we were to perform this analysis, what we would end up finding is so each of these dots represent bacteria, and then we're looking at both characteristics for those bacteria. So for example, say for this particular dot right there, we're kind of saying that its DNA hybridization is, above 25%, less than 50%, it's probably like, 35%, let's just say. So that means not the same species, but potentially the same genus. That's kind of what we would say. As far as the sequencing goes for the same organism, it's only about 50% though, overlap. All right, so it's definitely, again, not the same species. Where we get into some kind of problems, or at least confusion, is you might say, okay, well, what about this one up here? Okay. Now, as far as the sequencing goes, it is over 97%, But as far as the hybridization goes, it's really low. Right? So what we're going to find is that there's kind of this zone here. Okay, so the 70%, 97%, so 97% sequencing overlap, 70 greater than 70% hybridization. These we're definitely certain of using two types of analysis that they are going to be the same species, right? These others, then we start to say, oh, if we only did one technique, we may say yes, they're the same species, but if we only did the other technique, we would say no, they're not the same species. The point is, it's always best to do multiple types of analysis, right? And to either find agreement between those analysis or a disagreement, and then other factors would have to come in to make the decision. So potentially some of the more traditional methods on classification might say, well, this one tells us that they're the same species, this one says they're not, but we also know these other characteristics of the organism that may then push it one direction or the other and say, yes, they are, or no, we're not going to support that they are. So those are some of the ways that this type of analysis is then interpreted. There's another type of analysis that I'll bring up here, and it is used more to kind of look at organisms to say whether they're really part of the same group or not, and we do use it quite a bit in the breakdown of the gram-positive bacteria, and that's this one here. The only one I haven't really talked about yet is a GC ratio. Kind of erased my drawing here. So what we're doing is for a particular bacteria, obviously the C's and G's are going to pair up. So it's going to be the same percent depending on which strand you're looking at. So let's say of these here, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And of those 10, I have 1, 2, 3, 4, 5. Okay, so 50% is GC. So these are these here for this particular little sequence I have. That is something that can then be used also to tell the difference between groups of organisms by looking at just the percentage of G's and C's. This is not going to be the same. It's not always going to be 50%. It might be 20%. It might be 23%. It might be 70%. Different organisms are going to have different amounts of the G's and C's versus the A's and T's. It is not an exact equal amount. It's one of the things that makes all the organisms unique and different. Typically for gram-positive bacteria, after finding a cell wall, whether they're gram-positive or not, really the next characteristic that is looked at beyond gram stain then is GC ratio. And so we have high GC and then low GC. So high GC is greater than 50%. Low GC is less than 50%. What if they're exactly 50%? Then other things will kind of come into it to go, but for their characteristics. So this is just yet another thing that you will see is a characteristic of organisms and how they might be classified is looking at this, and that's essentially what it means. It means you're just looking at the DNA, counting the guanines and cytosines, getting a percentage, and that could put them into one category or another. And then for organisms, let's say we know that a Serratia has a certain typical percent GC ratio. If we see something that's well out of that range, we just know it's not Serratia. We don't know what it is necessarily, but we know it's not that particular organism. So you can get kind of spans of the GC for a group of organisms, and if there is overlap, say, oh yeah, this one's 43%, and this one's 44%, and that's within the range, then maybe they're the same organisms, then you then need to do different types of analysis to really figure out what they are. So, classic analysis, biochemical, morphological, and gram stain. The more modern molecular analysis still uses Gram-stain, uses DNA sequencing, uses DNA hybridization, uses the amount of guanine and cytosine in the DNA. And there are different rules. Make sure you know the rules. So for, this is the hybridization rules. Hybridization rules have to be greater than 70% to be the same species, greater than 25% to be of the same genus. Okay, and remember a couple little tips on this. For example, Staphylococcus aureus, genus must be capitalized, species must be lowercase, and they either need to be underlined or written in italics, or else they're not genus and species, because these are Latin names. So there's a couple rules about just the names themselves, how they're assigned, whether they're put into a particular group. Just make sure you're kind of familiar with that. The next thing we're going to go into is just an overview breakdown of the names themselves for gram-positive bacteria, the high and low GC, kind of the categories that they're put into, the names associated with it. The gram-negative bacteria, the categories they're put into, the names of those particular groups. We're not going to go into all the subgroups and all the names of the organisms within the groups, but I'm just going to give you a real quick overview of those organisms.